All right, so Travis is on, everyone can see him, and he can't see us, but at the end we can engage with a bunch of questions, and I'm just gonna, and I think you guys can log on too if you wanna see the screen. Nathaniel, will you share the Zoom link? If anyone wants to actually log on live on your screen too to have a closer view and type stuff into the chat box, anything like that. But this is the Travis Stiles that I introduced in the last lecture. And I will let him talk about, you can see his company right there, which is a new logo, right, Travis?
Yeah, it's the new fanciness.
Okay. So go ahead and take over, and then at the end, if we have time for questions, we'll rejoin you.
All righty. I'm just gonna get it to go. All right. Okay. Hopefully the AV works here. This is weird. I feel like I'm talking to myself. But all right, cool. Nice to be here. I've spent a lot of time in the room that you guys are in right now with Mark and Courtney. Courtney wanted me to come today and talk to you guys a little bit about, because you guys talked a bit about nerve signaling and nerve growth in your last class. So I was going to talk today a little bit about nerve regeneration, because that's what my company does, is we try to develop therapies that will promote regeneration of damaged nerve tissue, both neurons and glia, and so the whole kind of kit and caboodle. So I wanted to start with a couple really stark examples just to lay the framework. So bear with me while we do a couple little videos here that I think you guys will like.
All right, hey Styles, we can't hear the video.
Oh, there's no audio for it?
Yeah, there's no audio on that.
So that's not super important. So this, you can hear me though, right?
Yeah, we can hear you.
So you can see, I'll just turn the audio down. So here's this guy sitting here, and you saw him with no arms, and now he's sitting here with arms. And here's him doing his rehabilitation. And what they're talking about here is how for the first few months after he had this surgery, where he had these other people's arms sewed onto his body, he couldn't do anything. And it was just, all of his physical therapy was passive range of motion. And this is showing that over time, the nerves from his body actually grew into the new arms. And now he's able to do this with someone else's arms attached to his body. So that's pretty cool. This is Felix. Felix has no arms and no shoulders. And so, unlike the guy over here on the left, who basically was an amputee above the elbow, Felix over here was lacking both joints. And this is him after his surgery, where he had bilateral arm transplants all the way at the shoulder. And I'll just show you this. Here's him after a couple of years of recovery. So I just wanted to show you guys, because I think that it's really important to kind of note that nerves in the human body can regenerate. I mean, it's pretty profound that not only can they grow back after having an arm chopped off and sewn back on, but it can be someone else's arm sewn back on. I find that fascinating. Now, let's look at that in contrast to a spinal cord injury. And I think this is the kind of the prime example of spinal cord injury. Oh, here we go. So here we have Christopher Reeve, the iconic actor that played Superman in the late 70s, early 80s, was kind of your quintessential manly man, actor, signifying strength and all that kind of stuff. And here's him in the famous helicopter scene in the Superman movie versus Christopher Reeve before his death. So Christopher Reeve was paralyzed in a horse riding accident from the neck down. And he spent the last decade plus of his life in a wheelchair, unable to move anything below his neck, and was on a ventilator, and never got better, despite his foundation, the Christopher and Dana Reeve Foundation, being one of the most well-financed scientific research groups in the history of the globe. He was never able to, find a way to restore any of the function that he lost after his accident. And he eventually passed away due to complications from his injury. It's been a while now. I can't remember the date. But that, the life expectancy for people who are paralyzed, especially quadriplegics, is diminished because of some of the complications that can happen from the injury. So why is this? So this is, again, just kind of showing you the kind of with a contrast between Christopher Reeve Superman, between Christopher Reeve paralyzed, to really reiterate, how devastating and irreversible these injuries can be. And on the right, and it's not super important for this lecture, but just showing you that not only is this like a really catastrophic injury in terms of quality of life, but they're really expensive too, with lifetime costs associated with injuries ranging anywhere from $1 million to $5 million. Now, the contrast that I've just tried to lay out for you guys is like, okay, nerves in your body can regrow so well that they'll actually regrow into other people's limbs if you put them on there, whereas nerves in your brain and spinal cord just can't. Well, and why is that? Is there a difference? And I bring up, so I have Imhotep on here because Imhotep's considered by many to be the first physician. And I think he was around in like 2500, BC. And he actually documented in some of his ancient scrolls or whatever, that he had actually documented the regenerative failure of brain and spinal cord injuries, and was the first to kind of comment on the regenerative capacity between peripheral nerves and central nervous system nerves. And in that idea, he actually postulated that the nerves in the brain and spinal cord lack the intrinsic ability to regrow. And if you think about it conceptually, that's not a crazy idea. If you think about like, what do brain and spinal cord nerves do versus what do nerves in the body do? Nerves in your body do heat, pain, temperature, proprioception, maybe some movement, right? Little muscle fiber action. But what do nerves in your brain and spinal cord do? Reflexes, they kind of is like the minimum function, but then you have things like language and dreaming and consciousness. And so the idea was like the complexity and the job. I mean, these nerves are just super specialized, and if you mess them up, there's no going back because it's too high of a bar, and that's it. And it wasn't until this guy, Dr. Albert Aguayo, and some other contemporaries that I'm not bringing up because he seemed to be central, decided that they wanted to test this theory. So we're literally talking all the way up through human medical history up until the late 70s, early 80s, where this guy decided, you know what, I'm gonna challenge this assumption. And so this picture you see on the right here is a rat brain. And what they did is, you can see the rat eyeball. I don't have the pointer going, so you guys probably can't see what I'm pointing at here. But you have this rat eyeball that normally goes into the brain here with the dotted line optic nerve, and they chopped it off. They took the eye, chopped off the optic nerve, and they took a graft from a sciatic nerve, sutured the end of the eyeball onto that sciatic nerve, and then looped the sciatic nerve all the way up through the skull over the brain and reattached it in the rear of the brain, because that's where the visual center of the brain is. Get a little drink here. And what they found was those nerves, because eye nerves are actually brain nerves, right? What they found is while those nerves, if in a normal injury, if they were to crush that optic nerve, wouldn't grow back, when they used the peripheral nerve graft, those nerves grew readily all through the graft. And the reason that was so incredibly cool and important is because it was the first time that somebody showed that there is an intrinsic capacity of these nerves to grow, but they won't grow in their own environment. But the peripheral environment is more permissive. And so this really is the paper that really started it all. I think this was published in '81. That was the first one to say that you get extensive elongation of damaged axons into a peripheral nerve. And they also did the opposite experiment. So this is a sciatic nerve graft. So they took nerves from a sciatic nerve bodies, and tried to suture them onto a central nervous system nerve graft. and they wouldn't grow. The nerves would grow right up to the interface of where the CNS graft was, and then they would stop. And so the kind of take-home message there was that it wasn't an intrinsic failure, it was more extrinsic. And the reason that's good news is if it's an intrinsic failure, if the nerves just lack the ability to regrow, then to try to promote them to regrow really would require changing the essence of what they were, changing their genes, changing things like that. But if it's an extrinsic factor, then you have the ability to potentially come in and block that extrinsic factor or overcome that signaling pathway. You have the opportunity to potentially reverse that inhibition. So just as a quick summary of like the differences. So central nervous system nerves don't regenerate, peripheral do, but those same nerves that don't regenerate will grow if you put them into a peripheral graft. And there's really two main factors that go on here. In the acute phase, so the first days to weeks, you have myelin debris. And I'll talk a little bit more about myelin. And then in the chronic phase, like longer term, you get like glial scarring. And at that point, you get what's called chondroitin sulfate proteoglycans or CPGs, and those will inhibit regeneration as well. So you kind of have the spectrum. In the early phase, you have molecules that are in CNS myelin that actually inhibit regeneration of nerves. And in the longer phase, you have these CSPGs. And just because I don't know how much you guys have talked about myelin and neuro, but basically when you think about nerves, you basically have you can equate them to an electrical wire. You have the metal part that can actually conduct electricity, and then you have the plastic coating. And that plastic coating doesn't just protect birds from landing on it from getting electrocuted, it also makes the transfer of electricity more efficient. And I know that Courtney in your guys' last class talked about how thinly myelinated versus heavily myelinated nerves can have different conduction velocities. It's the density of the myelin structure can actually improve how quickly signals are transferred. And that's one of the main things that's different between a central nervous system nerve and a peripheral is not just the thickness and the density of the myelin, but also the composition of the myelin. You can imagine that because the levels of activity that the brain does are so complicated that the myelin itself is also extremely complicated. And so when it gets blown up, there's a lot more stuff floating around. And so this is basically a diagram that breaks down that all of the and this isn't comprehensive, but this is kind of the layout of all the different molecules and how they work and how they kind of converge to end regenerative, to block the regeneration. So I want to point out, and I should have turned on my little pointer. I don't even know how to do that now. Can I turn this on here? I don't know how.
Stiles,
Anyway.
if you can hear me, your pointer is on. We can see it on the screen, the cursor.
Oh, sweet.
And
Great.
Van Ness had a question.
Yeah.
Travis, hey, in that experiment where they grafted the eyeball, could the rat's eye move?
Oh, that's a great question. No, I don't think so, because they had to cut all the muscles away to get the eye out to sever the optic nerve. So, but it did redevelop pupillary response. So if you shine a light on it, it reacted to light over time, which is nuts. That's just craziness. I don't think it saw things though. I don't think they regained vision. I don't know. What is that? Anyway.
One more question, Travis. Kevin, what?
For the sciatic nerve, was it because it was a sciatic nerve that was regenerating and they transferred it, or is it like the CNS, like the nerves don't connect and they don't regenerate because you use a different proliferator that was able to.
I'm hearing like every third word. Sorry.
Okay, well, let's come back to that one at the end. Okay. All right, go ahead and just keep going, guys.
Okay. Yeah, if you want to type your question in the chat, I can kind of circle back to it in a bit. So basically, so somebody say something? No? Okay.
Yeah.
So basically, in 1981, now we've got, 40 plus years of research now where people, like I said, okay, there's these external factors. 40 years later, we have a pretty good idea of what these factors are. So this top layer is basically, this is the components of myelin that we know inhibit regeneration. Here's some of the factors that we know some of the receptors that we know are involved in their signaling. But what I really want to point out here is this row A. one of the things that has really perplexed scientists is they've been able to prove MAG, Nogo 66, amino Nogo, ephrins, semaphorins, all these things. They've been able to show individually one by one that these molecules will actually stop nerves from growing. But the problem is they're all very different molecules. It's not common that you get different, like really different proteins that end up signaling almost the exact same way. And as people started looking at, okay, well, what are the receptors that they're interacting with? They started realizing, wow, even the receptors are very different. What's that? Is that me? No? So the heterogeneity of the receptor system made it difficult too, because if you block one receptor, like if you were to block this ephren 4A receptor, you still have Nogo receptor, PIRB, all these other receptors. So you couldn't really target one interaction to kind of restore the growth capacity, because there was these redundant signals that all ended up converging on this RhoA activation. And I'll point out, RhoA is not a bad guy here. RhoA is a super benign molecule that every cell in your body has, and it's part of a family of called small GTPases. And basically, there's these, and this is a oversimplification, but you can think of it like a construction crew. Rho, RAC, CDC42 are these three molecules, and they're like three important members of a construction crew. One of them's demolition, one of them's like scaffolding, and the other one's like moving stuff around, right? And so you'll never have demolitions and building in the same area. So they're very tightly regulated in terms of the spatial aspect of the cell. As the cell moves, the back end of it is going to want to be picked up and drug along, while the front of it is going to be trying to build and extend to kind of reach and grab. And so you'll have rack in the front active and row in the back active, so that it can kind of pull up the train tracks and then lay down new train tracks up front. So activating RhoA typically isn't bad, as long as it's really tightly controlled and it's only active where it needs to be. Part of the reason why this kind of really unique signaling event happens is that when these myelin proteins are just kind of blown up, in development, you can imagine as nerves are really growing and they're forming these really, specific structures, that what you don't want is you don't want them to just grow willy-nilly. So as the brain structures are defined, it will lay down little molecules like street signs saying, don't come this way, don't come this way, turn around. But when you blow all that up and all those cues are now just mixed up, now instead of getting really refined and specific signals, the nerve is overwhelmed. In my kind of, if I'm really to break it down, I actually think that the reason the nerves, they just give up because they can't interpret this overwhelming amount of kind of like contradictory signals. And that's why you get this regenerative failure. So this is just kind of breaking down one by one. So role of small GTPases, RhoA. The first receptor that was discovered was the Nogo receptor. And I highlight this for a very specific reason. So if you look at the black receptor over here, NGR1, this is what's called a GPI anchored protein. And the reason this is important is you need to be able to communicate to something inside the cell in order to get a signaling event. And the Nogo receptor doesn't have an intracellular domain. It just sits on the cell surface, which means it doesn't have the ability on its own to even cause a signal. And the fact that it was so central and it was characterized so early on was one of the first clues that the signaling mechanisms here were more complex than just a single ligand receptor signal event. So P75 is one of the most interesting ones here. So P75 actually doesn't bind to any of these molecules, but it seems to be very important for many of them in terms of signaling. So basically, if I'm going to oversimplify, P75 has a really important intracellular domain. And one of the ways that Nogo receptor binding to these myelin proteins can actually block nerve growth is because it also will just snuggle up to P75. And when they bind to each other, P75 starts to do things inside the cell that leads to that Rho activity. And that's what I highlight here with this NGR complex. So it's this complex that forms that actually leads to the Rho activity. And if this complex doesn't form, you don't get the inhibition of growth. So moving on, because this can get a little deep here. The key here that I want to point out is that for a long time, people thought that they couldn't recapitulate a regenerative response. Blocking, genetically silencing any of these proteins didn't seem to work in terms of getting nerves to grow again. So they started to think, well, we must be missing something. There must be another receptor. And so there was this dogma in the field, you know, for quite a bit, that there was just something that they hadn't identified. And this is not super. This is just kind of breaking down the Rho activation and some of the receptor complexes that can form. This is kind of just mechanistically showing you how the kind of different machinery comes together to activate Rho and block regeneration. But the kind of the main message here is, you can see in a lot of these, it's the combination of certain receptor complexes that leads to the effect. But you know, I know you guys talked about nerve growth factor in your last class, and nerve growth factor can actually bind to P75. P75 was actually originally identified as a nerve growth factor receptor. But when NGF binds to P75, it actually stimulates growth of nerves, not blocks it. Whereas when MAG or OMGP or some of these other ones bind to P75, or they actually doesn't bind to P75, but it binds to Nogo Receptor, and then Nogo Receptor binds to P75, that actually stops nerve growth. So that brings me to Novoron. So now I'm going to get a little bit more into what we talk about. Actually, we can take a few questions because I want to make sure that the background kind of mechanisms are understood. So with the rat eye nerve, is it because it was a sciatic nerve that it was regenerating, or would the nerve, if it was still the optic nerve, regenerate as well? So it wasn't actually the sciatic nerve, it was just sciatic nerve graft. So basically, a nerve has a cell body and then an axon. It's like, this would be my axon, and then my cell body would be, you know, my brain, my torso, right? So a sciatic nerve actually doesn't have any cell bodies in it. It's just the axons. So when you take a sciatic nerve graft, it's actually considered to be decellularized, which means there's no true nerves in it. There's remnants of nerve tissue of remnant axons, but because they're no longer attached to a head, they're not really nerves anymore. So when you sew that sciatic graft onto the optic nerve, all of the cell bodies for the optic nerve are actually in the retina. So all the cell bodies are still there. You've just chopped off their arms. And now, just like the kind of the arm transplant guy, you've taken the sciatic nerve graft and sewn it on there. And now the optic nerves are growing into the graft. Does that answer the question?
Nodding in the direction of yes.
Okay, I didn't hear that, but I'm gonna assume.
Yeah, it did.
Okay. So now we're going to talk a little bit more about Novoron. We'll talk about the science, but we'll also talk a little bit about the story, because I do think, and we might not be able to get to this, but I do think our story, Novoron's story, is unique and potentially interesting because, you know, Mark was my advisor at UOP. Courtney and I were grad students there at the same time. For me to go from there to having a startup biotech is not normal. And so if we have time, I can talk a little bit about that and answer some questions. So I actually, as you can imagine, you know, doing the sports sciences, physiology program at UOP to now running a company that does neuroscience is a bit of a weird path. So how did that happen? So I've always had an interest in neuroscience. On the left here is one of my undergrad fraternity brothers, Callan Morinaka. My senior year in college, Callan was diagnosed with Lou Gehrig's disease. And if you are familiar with that disease, then you'll know that the younger you are when you're diagnosed, the more rapidly the disease progresses. He went from being a junior national judo champion to passed away within 6 months of his diagnosis. And on the right is my uncle, Lenny, who was similar to Christopher Reeve, was paralyzed in a horse riding accident, actually a horse racing accident, in his early 20s. And he passed away recently, 5 years ago, from a very treatable form of bladder cancer, which is another example of how life expectancies can be shortened in paralyzed patients. Because this particular form of bladder cancer is usually detected very early, because for non-paralyzed patients, it hurts. You experience pain, and so you get early detection. But in a paralyzed patient, he didn't realize he was having problems until the cancer had progressed. So I've always had an interest in neuro. And I always make sure when I'm giving these talks that I always bring up both Courtney and I's advisor from undergrad, Julianne Abendroth Smith. So the reason I bring her up is because literally I wouldn't be here without her. I was not a dedicated college student. I was pretty good at being a college student and kind of liked it too much. In my senior year, I'll never forget, she called me into her office and she's like, oh, hey, you know, you're going to graduate. What are you going to do next? And I was like, I don't know, maybe I'll stick around. Maybe I'll be a grad assistant coach for a while. Who knows? And she was like, absolutely not. You're going to grad school. And I was like, okay, well, application deadlines are passed, so I'll go next year. And she was like, no, what if I found a program that would take you now? And I was like, you mean like, so I don't even have to apply? And she's like, yeah. And I was like, well, that would be great. Appreciate the offer, but I don't want to pay for it. I don't have money for it. She's like, what if I also found you one that would give you a teaching assistantship? And so she actually found me, and I believe Courtney too, but I might be wrong, my teaching assistantship at UOP, and that's how I ended up there. So she leveraged her personal social capital to get poor Mark to take me under his wing and take a chance on me. And so I always make sure I give her her props, because unfortunately Jules passed away 10 years ago suddenly from a brain aneurysm, and so she never got to see the kind of the fruits of her investment into my laziness. So that brings me to the more relevant story to this presentation, now that I've given the kudos. So when I started grad school at UCSD, I was not familiar with the story that I just presented to you. I actually was still operating under the assumption that the spinal cord nerves didn't regrow. My Uncle Glennie couldn't walk again because those nerves could not regenerate. That was just what I thought. And I think a lot of people still think that. Binhai Zhang was one of the professors that was in my graduate program. And the cool thing about the UCSD grad program is that they don't let you pick a lab your first year. So typically a doctoral program in Europe, you don't even apply to a grad program. You apply to work in a lab that happens to be part of a grad program. And once you've done enough research, you get your PhD. At UCSD, you apply to the umbrella program. And then your first year, they make you do rotations in multiple labs and get exposure to multiple different types of research, so that when you do make a decision, it's a better fit. And you know your options better. And it was a really cool opportunity. And Binhai was in the neurology kind of unit was talking about spinal cord injury. And so he was the first person that really brought up this idea that spinal cord nerves can regenerate. He basically gave you the whole story I just gave you. It gave me the whole story. And I was like, wow, that's really cool. So if you could block these inhibitory factors, you could really do some things. You could make people walk again. Fast forward a couple weeks in the same session, Katerina Kosoglu, who is a extremely well-known scientist in multiple spaces, but particularly in the multiple sclerosis field. She was actually one of the first scientists to really prove that multiple sclerosis could be a consequence of a demyelinating event first that would lead to an autoimmunity, because most people at the time thought it was an autoimmune disease and that was the end of it. But she was actually one of the first people to show that you could actually get MS before you triggered autoimmunity. And she was talking about, multiple sclerosis is basically you get these attacks, these lesions in your brain, immune cells attack your own myelin. And then once a cell doesn't have myelin anymore, the cell gets sick and dies. She was actually, pointing out that like part of the reason that MS patients progress and eventually, pass on is because these lesions, once they form, they don't heal themselves. The myelin doesn't repair itself. And a lot of the reasons why that happens is the same for why nerves don't regrow. The molecules, the signaling, they overlap a tremendous amount in terms of why myelin doesn't repair in the brain as well. So the cool take home with that was, wow, so if you found a way to make nerves grow again, if you found a way to kind of block these signals, not only could you potentially make nerves grow again, but you could actually, reverse this damage in multiple sclerosis as well. And then like 3 months later, I'm in the oncology unit, and I was not interested in oncology. Everybody does cancer research. Probably, 3 out of every 10 scientists probably are doing cancer. So I was listening to this presentation from Steve Gonius, who's the chair of pathology at UCSD, and he was talking about inflammation in cancer. Really cool stuff, but just wasn't like my interest until he had his very last slide. I think it's here, where he was talking about how they had discovered a new receptor for myelin debris. And his literal comment when he showed this was, we don't really know what to do with this. We found this, the paper seems to be popular, people like it. So I wanted to show you, but I don't know what's going to happen. And so I was like, I was immediate, like, you know, are you aware that the whole kind of concept in the spinal cord field right now is like, there's a receptor that hasn't yet been identified for myelin proteins that might be responsible for regenerative failure of neurons. Have you looked at this to see if that might be playing a role? And he was like, no, and we're not going to. And so I actually approached him after the class and I was like, can I rotate in your lab and just try this? Like, just see if this is playing a role. And he was like, nope, hard pass. I don't want to do neuro. Thanks. Nice for meeting you. Nice meeting you. And I was like, okay. So I actually went back to, what's my next slide here? So I actually went back to Katerina and Binhai and was like, hey, have you heard of this? And they were like, no. And I was like, well, can I rotate in your lab and test this thing? Binhai was a brand new faculty, and so he didn't have the money to take me on. And Katerina had just gotten a job at the Gladstone Institute in San Francisco. So she was like, you know, if I'm gonna do this, you have to come with me to San Francisco. And I don't know if you guys have been to San Diego, but that would be a downgrade going to San Francisco from San Diego. So I passed. But they both agreed that it was a cool thing, that it potentially was important. So they marched me over to Steve's office and said, you got to take this kid. You got to just try it. Like, we'll mentor him. We'll serve on his thesis committee if it works out, whatever. But, you know, this deserves to get checked out. It's like, you know, a rotation is between like 6 to 12 weeks. So if it doesn't work, move on. But if it does, it's cool. So he agreed to let me try. And then, so the whole idea was that nerves don't typically grow in the presence of these myelin proteins. And so I wanted to see if you blocked this receptor, this LRP1 receptor, would they then be able to grow again? So how do you look at nerves growing? So I'm just going to show you a couple different ways of how you look at it. So these are nerve cells over here on the left, and you might not be able to see this. This is nerve cells. I believe these are PC12 cells that are stimulated with NGF. And you can see that they just start spitting out spider webs and connecting with each other. And they'll actually go and they'll connect the arms of different of other nerves. They'll form little synapses and you get this cool little network of nerves. That's one way. This is actually super cool because this is actually in a developing rat. And these guys actually made a fluorescent axon where you can actually see these are fluorescently labeled neurons growing through rat tissue. So these are like in an actual animal. And you can see they know where they're going. And these are actually optic nerves, retinal ganglia cells going back into the brain to the visual center, right? So these are like, there's ways that you kind of measure how nerves grow. So I give you that kind of background to then bring up my very first grad experiment. So I took nerves, same type of nerves that you just saw before, and I grew them in a normal kind of like happy environment. And you can see here that these nerves are sending out really pretty neurites. They're happy, they're surviving, they're sticking to the plate, they're doing a great job. In my next experiment, I took purified CNS myelin. So myelin, remember, myelin has all those different components that signal to stop nerve growth. So when I put those same nerves on purified myelin, you can see they can't really send out as many axons. They bunch up, they're not happy, they don't like to sit down, and they're just not doing as well. But when I used an antagonist to block that LRP1 receptor that they had discovered was a new receptor of myelin proteins, those same nerves grew readily across this substrate. Now, so the thing I want to emphasize here, the dogma was nerves can't grow on myelin. And as you can see, not only were they growing on myelin, but some of these axons would actually burrow under the myelin and just kind of figure their way through. And so I was the luckiest grad student in the world because my literal first experiment, after a good long time of being trained and learning how to do these systems, I basically proved that this receptor was involved in regenerative failure. It's the luckiest thing. Don't get me wrong, the rest of my thesis work was not as easy, but I was very fortunate to like right off the gate know that I had something. So again, I just want to taking a little bit more simple overview. So you have the myelin debris here on the left, magum, Nogo, different things. And then you have the glial scar over here on the right. And basically whatever the receptor complex is, it leads to ROA activation and that leads to regenerative failure. But the thing is I want to emphasize here the two outcomes in a damaged brain are failure of axon regrowth and failure of the repair of the myelin sheath. Here's a little summary of LRP1. This is what we work on. We work on targeting this receptor now to try to promote nerve growth. This is not, there won't be a test on this. It's a massive protein. It's a member of the low density lipoprotein receptor family, and it's largely considered to be like a scavenger protein or a lipoprotein receptor. Its role, even though one of the tissues that's the most highly expressed is brain tissue, its function in brain is not well understood. So, and I'll try to go through this pretty quick. So like the whole idea is what makes LRP different. Other receptors had been found that were known to be involved, but nobody had been able to show that if you blocked one receptor, you could make nerves grow. Whereas we were showing that if you genetically silenced that receptor or blocked it, you could overcome the inhibition of a variety of molecules, which was super promising. And so the idea was like, why? Why is this different? And so I wanted to both validate this as a therapeutic target, but also understand why it was different. And so the questions that I asked in my thesis work is, okay, is this receptor not just binding these proteins, but is it also facilitating their uptake into cells? And basically going down the rabbit hole of like the actual nitty gritty of how this signaling event happens. Is LRP1 responsible for it? Now, so part of why that's important is because normal cell signaling is very well controlled. So you get a signal that happens, it does its job, and it gets turned off. Part of the reason that you get chronic failure of nerve regeneration is that Rho signal just keeps going and keeps going and keeps going, and it never really turns off. And part of the reason for that is because you get these signal zones. So instead of a receptor getting turned on, doing its job, and then getting turned off, you get these vesicles that form that literally just keep turning on and turning on and turning on the Rho, while this vesicle, instead of getting trafficked somewhere where it gets broken down or turned off, it just goes all the way from where it got turned on all the way back to the cell body, turning on Rho molecules. And that's a long ways in some nerves. So it can be doing that for weeks, just turn it on, turn it on, turn it on. And so basically this was my thesis work. You know, does LRP1 internalize myelin? Yep. This is, I won't get too far into the weeds here, but basically showing RAP is an antagonist of LRP1, blocks its function, or genetic silencing of LRP1. Either way, you block myelin proteins from getting taken into nerve cells. When LRP internalizes this stuff, when that happens, does that internalization vesicle look like a signalosome? Because signalosomes are different than other normal functioning like vesicles, like lysosomes. And so what we found was yes. So over here is a little diagram of like what a signalosome looks like, RAP5 positive, containing LRP, containing P75, things like that. So we just showed that yes, the LRP dependent internalization leads to signalosome formation. And then the Rho A. So this is the big one, right? Because Rho A is where all of these signals converge. And so in theory, if you block the Rho A hyperactivation, that's it, that you should be able to make the nerves regrow. And so here we show that in cell culture, so over here on the left, these are just nerve cells, that yes, we can make the Rho signal go away after treatment with a variety of these myelin proteins that we know block regrowth. But over here on the right is probably one of the most important studies I did in my academic life, where we actually showed that this doesn't just happen in a dish, but we actually can make it happen in an animal. So we took an animal, we gave them a spinal cord injury, and we infused it directly into the spinal cord, either like a vehicle, like a saline control, or an antagonist of LRP1. And after 5 days, we went in and we looked at the Rho activity. And we showed a dramatic decrease in the Rho activity when we infused these LRP1 antagonists into the spinal cord. And that was really the start of when we knew that we might have an actual ability to make a therapeutic. And this is just showing in vivo that a lot, even in vivo, we were able to see a lot of these mechanistic events that had been well established in vitro, such as P75 associations with LRP1 and things like that. We were able to show that that machinery was also involved with the LRP1 signaling. And then this is just showing that the P75 association was also involved with Rho. And then this is just showing, because one of the things I had to do is in 35 different ways prove that blocking LRP1 or silencing LRP1 made nerves grow. So this is just a handful of different ways. PC12 cells, primary cerebellar granule neurons, dorsal root ganglia, retinal ganglial cells on purified myelin proteins. Every which way I had to really just cover the gamut to show that LRP1 was different because it mediated multiple different types of inhibitory molecules effects, which is what makes it unique. And so this is kind of the summary. So the takeaway here is like what makes this unique is that all of these molecules over here have nothing to do with LRP1. So P75, LINGO, NGR receptors, whatever we are talking about, myelin proteins can bind to these and they will form a receptor complex. If LRP isn't there, that's the end of it. They'll bind, they'll form their little trio or whatever kind of combination it needs to be that we know is necessary. But without LRP1, no Rho activation happens. When LRP1's there, it actually grabs the whole thing, pulls it over into a different section of the plasma membrane. And that section is where the signalosome is formed. And that's what leads to the chronic hyperactivation of Rho A. So the reason LRP1 is central, and this goes against the kind of original paper, is not because it's actually binding to the myelin proteins. That was kind of an oversimplification. It's because it's binding to the other machinery that's binding to the myelin proteins and leading them to where they need to be in order to have their signaling event. And that's really the secret sauce. And that's what was the complexity of this project and what took us so long to really hammer it out. So now we're going to find out if I talk too much. Courtney, do I have any time left or should we get to questions?
Yeah, use that time. We have half an hour left in the class.
Oh, sweet. I mean, so when I was watching your lecture off your website, you went like 21 minutes and I was like, oh man, I got to move.
Yeah, that was the students presented first and then I presented last for that one.
Cool. All right.
We got half an hour.
So I was flying through a lot of that stuff. I apologize if it was nonsensical because I was trying to like not take up too much time. But now, so we'll talk about like, because this is what I'm passionate about, right? So I was a puny little grad student that stumbled upon this really cool thing, ended up doing a lot of work trying to characterize how it works. And at the end of my thesis, I was like, there's a really good chance here that we've discovered a way that we might be able to repair nerve damage, make people walk again. That's really cool. I mean, most scientists get into science because they want to do something like that. Like basic scientific discoveries, you know, like a lot of the really impactful stuff, nobody will ever really hear about because it's like the foundation to the foundation to the foundation to the next drug or device or whatever. And it can be really important in the scientific community, but it's many orders of magnitude removed from actually helping people. It's not very often that a puny little grad student stumbles upon a project that has potentially direct relevance to a disease state. And so my boss was actually like a really prominent scientist in the cancer and LRP space. And we built a bit of a team around my project. Like once we got some traction and we realized what was going on, we added some scientists around me. We brought in this staff scientist, Alban Gaultier, who ended up kind of being my mentor because he had more neuro expertise. And we did a lot of great work. But as I got closer to graduating, the success of the project led to some of the team we built getting jobs other places doing cool stuff. And so when I was getting close to graduating, basically there was only 2 of us left. And my boss basically came to me and said, you know, like, I'm still a cancer guy. I'm still not a neuro guy. So like, once you're gone, I don't really have the ability to backfill and keep this project going. So I'm not going to keep working on this. So you're free to take it with you, which was a kind thing to say. That's not very common, right? But the problem is that it's really hard when you're just graduating with a newly minted PhD to go interview for jobs and be like, hey, do I have a project for you? I know you're interviewing me for your thing, but let me just convince you that you should do my thing instead. That doesn't go well. They don't love that when you tell them what you want to do instead of saying you're going to do what they want you to do. And so I found myself in this really difficult position where I was too green to have my own lab, but I couldn't find a lab that was willing to take me on and let me work on my own stuff. And this, by the way, this is Steve here on the left. This is Alban, I was just mentioning. He actually got a faculty job at UVA and still collaborates with us pretty heavily. But it was kind of his departure that kind of, you know, put the nail in the coffin on Steve being able to keep doing the work. So a couple friends of mine, Sean and Taylor, said, you know what we should do is we should just start a company to do this. Like we should just, because you guys probably don't know. So the National Institute of Health is like by far the largest funder of scientific research in the world. And the Small Business Administration, like in the 90s, I think, said, okay, there's a lot of really good academic scientific discoveries out there, but not very many of them are actually getting taken out of universities and into companies where they can become new technologies and things that actually change lives. And it was because there was this bottleneck, like there wasn't any money to do that. Like you could get grants for basic science stuff, but once you started a company, you had to raise money, and it's hard to do. So they created a program called the SBIR program, the Small Business Innovation Research Program, where Congress Congress actually passed a law where at least, I think, 2.5% of all research funding had to go to translational research. And if you're not familiar with the term translational science, you basically have basic science, which is like discoveries and new proteins and new mechanisms and all that stuff. And translational science is taking those findings and turning them into products, drugs, devices, whatever it is that actually impacts the world around us. And so by designating funds to do this, it's actually been an incredibly successful program. More technology is leaving universities, more innovations happening. And so it gave us the opportunity where we could actually write grants as a company to get this thing funded. But I, as a newly minted PhD, was like, there's no way. I don't know how to start a company. I barely know how to do science still because I'm still a science baby. Like, this is terrifying. So my reaction was not great. I did not want to do any of this. But this is another kind of thing that is not super relevant to the topic at hand, but I feel passionate about. Part of the reason that I felt that way is because I didn't feel like I was the right person to do it. I felt like there's so many people who've been working on spinal cord injury that for me to then say, I'm going to start a company because I'm smarter than everybody else and I can cure this condition and nobody else can, felt like just an absurd statement. And so I didn't think that, I thought it was going to be a disaster to do it. But the reason I bring that up is because after I was pestered into actually doing it, is I've learned along the way that that is such an overly reductionist, simplistic way to think about it. First of all, everyone feels that way. And Courtney can probably back me up on this. When you get into your PhD program, especially at a place like UCSD that everybody's like super alpha, everybody will posture like they know that they're the smartest person in the room, but like everybody does that because they're secretly afraid that they're the dumbest person in the room. And so it's all this like feeling like fake it till you make it type of stuff. And so like those initial kind of reactions that I had to like, starting Novoron, what I realized is like, I'm definitely not the smartest person to ever do it. There's a lot of factors that go into being able to do what we're doing. And I think if I'm being honest with you, one of the factors that makes me unique is just that I'm super obnoxious and don't quit. And once I realized how cool this could potentially be, I literally couldn't handle the idea of letting it die and never knowing if it could have made a difference. So the persistence, the willingness to just be poor a little bit longer and do that kind of thing is actually like, those things are just as important as they are to having a good idea. So the reason I bring that up is because like going back to Jules and her making me go to grad school, like I never saw myself as a scientist or an academic or somebody who could discover a new drug to treat paralysis. I was a kid from Idaho that, happened to be, decent enough at sports that I could go to college and do that for a while. I never really thought of myself as like being smart and making that a career, right? Thought I would get a normal job, maybe go to med school if I was doing really well. But here I am. And like my work was successful. It's been cited and published and all that kind of stuff. And now we've got a company where now we've just hired 3 new scientists that are going to help us kind of advance this to the clinic and be able to take a drug into the clinic. So that I thought I would talk a little bit more about that process, because here's the thing, like one of the reasons it was so scary for me is because I had no idea what it would take. I didn't speak the language. I didn't have an MBA, et cetera, et cetera. So I knew the science, but I felt like there were so many other aspects of starting a company that I didn't know anything about. Intellectual property. Like when I was in grad school, part of the reason my project got as much traction as it did is the university kind of caught wind of what I was doing, and they kind of swooped in with their tech transfer office. Tech transfer is just basically like patent office. And they came in and they said, hey, we'd like to patent your project. I'm like, oh, that's cool. So I helped them like write a patent, but I had no idea what that meant. I had no idea what the different steps of writing a patent were, like a provisional versus a PCT versus all these different stages. Which, by the way, like just as a brief overview, if you have a good idea, the first thing you do is you file a provisional patent, which can literally be one page with a basic description of the idea. That lasts 12 months. By the end of that 12 months, you have to actually write what's called a PCT, which is a basically larger patent that actually incorporates all the things that you want to be able to do. And then that has 18 months. In that 18 months, that gets reviewed by patent offices, and they decide whether or not your claims or what you say is novel and protectable. And if they say yes, then you have what's called the international stage. And that's where you actually then have to file to get a patent in different countries, because different countries have their different patent offices. And that's where it also gets real expensive. And then if you do that, you have 12 months of the international stage review, and that's when you can get a patent issued. Novoron has, I think, 3 patents issued, and then a handful more that we put in. My very first patent that I did with UCSD got issued in like 2017, I think. And it was a big deal. Like people file patents all the time, but to actually get it approved and like countries protect it, it's not as common as you might think. Problem was, so I had invented this technology while a grad student. So the university owned it. I didn't. So even though I invented it, it was not mine. So when I started Novoron, I actually would go back to the university and say, hey, can I have my own technology now so I can start a company with it? And I had to actually give them money to license my own invention away from them so that I could then develop a company or start a company and develop a drug around it. And then again, how do you raise money so you can do that? So fun fact, science is super expensive. All of my friends that have done like tech startups, I secretly hate them all because they can be like, yeah, we had 20 grand and we sat in our garage for 6 months and we have a company. I'm like, 20 grand is like maybe 3 weeks worth of a burn rate in a life science lab. And so I mentioned before that you have non-dilutive types of funding like grants versus dilutive types of funding, which is, venture capitalists, private equity firms, angel investors, friends and family, all that type of stuff. But regardless, raising money is hard. That's probably one of the hardest parts. Unless you know a lot of rich people, which unfortunately, that's how a lot of startups happen, is they're just people that know rich people. And so they have proximity to cash. Then there's like facilities, like being at a university, you just had access to all the facilities in the world. But when you spin out a company, you kind of got to go out on your own and figure that out. Team. So I'm like, that's one of the hardest things, is figuring out like, okay, so on a budget, how do you build a team that's going to do this crazy thing? And I'm actually really excited about the team we built now. We've got our third new scientist starting in a week. Namit and Emmanuel are already here, and we're going to do some cool stuff. I'm really excited. We finally have the ability to kind of like go and do the big innovative things that we've always wanted to do. And then the business model. Like the business model of things is actually something I think people don't necessarily think about. Like when I started Novoron, I was like, we're going to make spinal cord drugs and we're going to sell them to patients. That's what we're going to do. And that is not how it actually works. Like, learning more about the process a lot of it we learned in this I-Corps program that the NIH stole from the NSF, the National Science Foundation. It's like innovation boot camp, where they call them innovators, people that invent technologies, basically get a boot camp in business. And the reason I think that's such a smart idea is because there's a million MBAs out there, and an MBA education is great. An MBA prepares you to do a lot of different things. But if you have an MBA and you don't know the science, you can't run a science company. Alternatively, if you don't have an MBA, but you are the inventor, you're the expert in a field, and you want to start a company, you can learn enough of the business that's specific to you that you can run a company. And so the I-Corps program was really about giving founders the kind of basic tools they could use so that they could get a startup off the ground and be able to run an actual productive company. And then, it's, I think, again, going back to the original thing, is like a huge part of being able to be successful in starting a new company or doing anything crazy off the beaten path is just the ability to grind it out, the willingness to. If you believe in something, put the time in. So our first grant that actually was how we were able to start the company, our whole goal was to just use a known, we didn't invent it, it was a known antagonist. So an antagonist is a molecule that binds to the receptor and stops it from doing what it would normally do. An agonist is a molecule that binds to receptor and activates what it normally does. So blocks its activity or activates its activity. So RAP is a known molecule that blocks LRP1 activity. And so we actually wrote a grant just saying, we want to see if you can use this as a drug to make animals walk. And the best thing, like the aims, the approach was very simple. We were going to give this drug IV, and then we were going to see if the animals walked again. And while we were able to show that we could knock down row function, we were only able to deliver the drug for 5 days. So we never even got to the point of being able to see if the animals walked again. And the reason for that was there was a couple big problems. One, the grant we had submitted was for $700,000 for 2 years. And then this is why I will never be a Ted Cruz fan, because at that time, that was when sequestration hit, and they took our $700,000 grant and took it to $225,000. So cut our funding by 2/3, did not change at all the amount of work we were supposed to deliver. So we were super strapped budget-wise. And then it became apparent really quickly that while RAP was a really useful lab reagent, it was not a drug. And the difference, like what they don't teach you in grad school, is the difference between a reagent and a drug. Like things like formulation and stability and just kinetics and all this crazy stuff that I didn't know about. So it was a really, really rough spot. The other problem is while IV is a fine way to deliver a drug, we didn't know what the right protocol was, how much drug, how often. Like, you know, the limitation in a rat is you can't give them an injection every day because their veins are so tiny that eventually they'll collapse or will scar over. So you're very limited in how often you can give the drug. So we learned a lot from that first issue. We learned that while people didn't like doing more invasive means of drug delivery, like ideally we would have wanted to give the drug straight into the spinal cord, but the people reviewing our grants didn't like that. So we went to IV, intravenous, because that's what we thought they wanted us to do. And that was a mistake. In retrospect, just because the reviewers thought that was smart, the reality was in humans, we have the ability to do intrathecal drug delivery, taking a drug and putting a tiny little port into the spine and delivering the drug directly to the spine. And that is the reason why that's awesome or ideal, is because if you don't have to give the drug to the whole body, but you can just give it to where you want it to go, one, you limit the risk of potentially having unwanted effects in other tissues. And two, you dramatically reduce the amount of drug you have to give. You can imagine that if you gave drug throughout the whole body, you probably need the amount of it that gets to where you want it to go is probably like 1/10 of a percent, 100th of a percent. So you need to use like 100 or 1,000 times more drug to get to where you want it to get, where the concentration at the site you need it, than you would if you just delivered it straight there. And so we had to go to a ton of different surgeons and experts and have them validate for us that, yeah, you can do that. So that when we went back to the NIH, we were like, listen, I know you don't like this idea, but the people that will actually use this drug say that that's the way to go. So like, this is how we're going to do it. The other problem was like this RAP as a drug. A huge part of what we did, we spent over a year where all we did is mess with RAP in order to go from a reagent to a drug. And probably one of the biggest and most boring kind of outcomes or wins that we've had at Novoron is the day that we were able to show that we had made a version of that molecule that actually could be used in a rodent study. It was so boring because it was just, can we make the thing stay stable at 37 degrees for more than a few days? Oh yes, that was a year of work. Very painful time. And then formulation, that's another thing they didn't teach us in grad school, is like, you don't just put this in water. You don't just put a protein or a drug in water and then it behaves well. There's you know, there's all sorts of things you have to put in there to like make it more soluble and stable and buffer things. And so our new molecule, our new lead molecule is
I did.
Novo 118.
Yeah, I was able to order it. I got a computer also
Sorry,
through
I
there.
can't.
And
Somebody talking?
you know, I know you had the budget that you gave me. For
Hey,
the
is that a
computer.
manual?
It was about, I think, around a grand or something like that. I don't know,
Can I
Bill,
mute him?
but the monitor, so the price there was 180
Hey,
something,
Emmanuel.
and then so I thought it was below 200, but then it tax and so it came out to
Can
like
somebody
20
mute
something.
Emmanuel?
Yeah.
There we go. Okay, cool. Sorry. Anyway, so getting back. So now Novo 118 is our lead. This is just showing you, this is like so much work. This is just showing you that we actually made a drug, not just a lab reagent. So I won't get into the super specifics, but we got a nice dose response out of it. We were able to show selectivity in a cell culture. It got into the brain, which is super useful. And then now, okay, so the whole point, we spent years trying to make this work. And but the goal is to give it into a paralyzed animal long enough to see if we can reverse paralysis. And so this is the most important study I've ever done. So this was a study that we did in collaboration with the Miami Project to Cure Paralysis at the University of Miami, where we took rats and we gave them what's called a dorsal hemisection. So at the thoracic vertebrae, so mid-back, we basically gave them a contusion injury through their cortical spinal tract. We bashed their spinal cord with very specific force and consistency in order to induce paralysis. And what you can see here so if you look at these pictures over here on the far left, you can see that after these animals are paralyzed, their entire hindquarters can't move. So they literally drag themselves along by their front paws, and their back paws just get dragged across the ground because they can't pull their feet up. Now, over time, because it's a contusion injury, not a transection injury, so we're not cutting the cord, we're just hitting it, the animals will recover some. So you can see, even in the vehicle treatment, you can see that the animals are no longer dragging their feet, they're kind of pulling them up to the side, but they still can't walk. They waddle, they kind of shuffle themselves along. Their tails they lift it up, they're still dragging their bellies across the ground, but they'll kind of use their back feet to scoot. But on the far right, these are the animals that we treated with Novo 118. So this is 6 weeks. We gave them 5 weeks of drug, and this is what happens at the end of 6 weeks. You can see that not only do these animals no longer drag their feet, but their feet are pulled up underneath them all the way, and they actually take steps. They walk. And the data to demonstrate how the difference there is over here on the right, where you can see the red line is the motor function with drug treatment, and the black line is just the normal recovery. And there's a couple things I'd point out here. One, after about between 5 and 7 weeks, the improvements in the control animals plateau, and you can see that happening here. Whereas the improvements that are the treatment animals is still going up. So we actually are doing studies now where we're going to go out longer to see how much better these animals recover, like because the recovery is still ongoing. And by the way, this scale is a 13-point scale. So the treated animals are at about a 9, and the vehicle animals are at about a 4, 4.5. And just for reference, you can see down here at the bottom, so people can see like what this scale is. This table down here shows you exactly what the scoring is. So you get you're measuring paw position. So like if their paws are being dragged, pulled up, or completely underneath them, if they're taking steps, so like clearing their toes when they walk, or just kind of like shuffling, and then their tail, if their tail lifts up and down, and you score based on those things. So you can see like a 9 is an animal that's like clearing the toes most steps, and like the paw position is good, but maybe it's not clearing the steps every time, and maybe the tail isn't always up or down, or the trunk stability is not great. The thing on the left here now that I clicked past the pictures, we don't know why this is happening, but one of the things that happens in human spinal cord injuries is after the injury is the acute events are kind of over, you actually form a cavity. You get cavitation, which is like this fluid-filled sac. And as you can imagine, if a nerve is trying to grow, it's like trying to rebuild a road and hitting a canyon. It's really hard to build a road over empty space. And so one of the interesting things we saw with Novo 118 treatment is that cavitation was reduced when we used Novo 118. We don't know why. We're still trying to work through that. We're working with Jay up here on the right to try to figure out what the mechanism is behind that. Completely unanticipated finding, but it's cool. So anyway, we've also have some other programs. We've got a multiple sclerosis program because, as I mentioned before, the same mechanism that we hit for spinal cord injury is involved with the failure to repair the myelin sheath in MS. And so this is just showing that in an animal where we had knocked out the receptor LRP1, that we actually dramatically reduced disease severity in an animal model of MS. And then this is another one, a Cooper zone model this is the one we did with Alban, who's now at the Brain Immunology and Glia Center at the University of Virginia. And then this is just showing that this is actually a predecessor of Novo 118, Novo 117, showing that in cells, we can actually, in a dose-dependent fashion, promote more myelin protein expression using our drug. And this is just some other data that we've done. So like we talked about optic nerves and, showing that when you do an optic nerve crush that you treat with our drug, you actually spare a lot of those neurons, they don't die. And this is our newest program. So we've been developing LRP drugs for a while, and it was recently published in, I believe, Nature, that LRP1 is the primary receptor involved with tau spread. So we don't need to get too deep into this, but Alzheimer's disease has kind of two core pathologies. You get amyloid plaques and tau tangles. It's this like protein aggregation that's like a hallmark of Alzheimer's. And tau tangles are formed as a consequence of this kind of dysregulated tau being spread throughout the brain. And it doesn't spread in like blood or CSF. It spreads because neurons that get too much of it actually spit it out and through synapses, they'll pass it on to other neurons. And then it spreads throughout the brain until it aggregates too much. And LRP1 is central to that. So if you block LRP1, you can prevent tau spread. So our newest grant that we got from the National Institute of Health is on trying to develop drugs to block tau spread as a means to treat Alzheimer's. So that's the rest of this is all kind of, company specific stuff. So I will stop there. And if you guys have any questions, happy to answer them. I'm going to stop sharing now.
Hey, Stiles, can I ask you a question real quick?
Yeah.
How far away from human trials are you?
So as a general rule, so you have kind of like the general drug development kind of stuff. So you have like discovery, which is we have a receptor we can target. Then you have like the development phase. So that when at some point you need to do kind of like pre-clin development, non-GLP. So when you hear people talk about non-GLP, preclinical non-GLP, it's basically all the stuff proving that it works in an animal model, whatever drug it is, getting all the data you need in order to have a compelling case. The reason I delineate and specify non-GLP is because at some point you have to make a GMP quality drug. And that is a big barrier to getting into the clinic, because GMP manufacturing, you have to pay somebody with a super fancy, basically drug making factory to make your drug in a super clean facility with people wearing hazmat suits and like having to use badges to get in places, because that's how we make sure the drugs are clean and good and well manufactured. Then you do what's called IND enabling. So you basically do all the non-GLP stuff to get all the data you need, both in terms of like, what is there amount of drug that's toxic? Is there amount of drug that is, what's the highest amount of drug that you need to make it work well? And then once you manufacture a GMP quality drug, you go into IND enabling. And the dumb thing is you spend all this money to make a GMP drug just so you can go back and redo all the things that you'd already done in a non-GLP fashion. And once you get all that IND enabled data, then you file what's IND, an investigational new drug status. And once that's filed, you can get approval for clinical trial. So we're basically at the stage right now where we're finishing up the non-GLP stuff, and we're actually developing our GMP protocol so that we can make a GMP drug. And we're hoping that we start that in about 9 to 12 months. And then we'll do our IND enabling stuff. So if all goes well, we're hoping that within 2 to 3 years, we can file that IND and be looking to get into human trials.
When does the classification of the drug start? Like as a spinal cord?
I can't. You feel free to yell or turn your microphone on, whatever.
All right, you can feel that.
when does the classification of the drug start? As like a spinal cord treatment or a spinal cord injury treatment or Alzheimer's or?
So basically, like when you like apply for a clinical trial, you specify what the patient population is, and then if your trial population works, that drug is then approved for that disease. So like in cancer drugs, you'll have drugs that start out and they'll be in a clinical trial specifically for one type of cancer. It'll get approved, and then they have to go back and do another trial all over again for the next type of cancer. So the indication that it gets approved for is really when you file that IND. Does that answer the question?
Yeah.
And then there was a question in the chat here. Are LRP, so one, RhoA is an inhibitory receptor. So no, RhoA is a signaling molecule that the receptors activate, and it's what it, so it's inhibitory when it's hyperactivated. Not its normal activity is fine, but hyperactivation leads to failed regeneration. Somebody asked, are LRP receptors located in other cells of the body or only the CNS? So the two tissues that express the most LRP1 are brain and liver tissues. So, but LRP1 is expressed in a lot of tissues. It's certainly not just brain specific, which is another benefit to just being able to do direct delivery into the spinal cord so that we don't risk mucking things up in other tissues that don't need drug. So, any other questions?
I'm not seeing other questions. Cameron has one. Okay, she's gonna come up and shout.
Yes.
Cool.
Since BDNF or exercise promotes BDNF signaling in the brain, would it be possible to combine LRP1 antagonist plus the combination of exercise to increase that myelin healing from the BDNF?
So that's a really good point and question. So like one of the things that we know is like anything that we're going to do to promote growth is going to be impaired if we don't have a really rigorous physical therapy approach.
Mm-hmm.
And it's specifically for the reason of like what you just mentioned and the other benefits of exercise. BDNF is one really important factor, but also just restoring the plasticity. So like, the issue here is like, this is your nerve, signal A goes to consequence A, but when you cut that and the nerve grows back, it won't grow to the same place. So even like when that guy was talking about, when I showed you that video, when the nerves finally grow back, he has to relearn because all those nerves that are now going into new places, the nerve, like, so if you're paralyzed and you regrow the spinal cord, the nerve that used to wiggle your toe might now make your knee itch. Like, so your brain has to relearn all that stuff, and it won't if you don't challenge it, if you're not pushing through. And BDNF is involved in some of that as well, but so there's a lot of other factors that come into play as well that exercise stimulates. And even simple things like getting blood flow to the distal regions of the body. One of the things that the Reeve Foundation actually kind of pioneered is one of the things that we now know can actually improve some outcomes in patients is hooking them, like patients that can't move their lower body, hooking them up to a bike and putting electrodes on their legs and making them pedal so that their body is actually doing that. There's actually some regeneration that actually goes backwards so that you can actually get some of those, some of that afferent function to come back and meet in the middle and maybe restore some function that way. So there's the benefits of exercise and rehabilitation are of profound importance. Like one of the things is, in designing a good clinical trial is you really need, like, so in identifying clinical trial sites, places that you're going to actually pay money for people to get your drug and to get treatment. One of the things that's going to be really important when we're thinking about that stuff is we're going to want to make sure that those facilities have top tier rehabilitation centers so that we make sure that our patients get really rigorous physical therapy, because if you don't do that, you're going to dramatically attenuate whatever the potential benefit you could have. That signal is going to be less. And the biggest thing in any clinical trial is you need to be able to show a signal of efficacy that's above the noise. Because when you have different patients that have different levels of injury and different levels of trauma, the outcomes of the drug effect is going to be very, very different because they started in a different place. So it's really important to make sure that you're getting the most signal out so that you can you can tease out the kind of normal variance in patients and get in and see real significance. So yeah, that's the short answer to your question is yes, that's super important, but it's actually like incredibly central to how you would be able to prove a spinal cord drug works.
Thank you. I have one last question. I know other people have to go, but were you having the rats were you monitoring how much physical activity they were doing?
No, so that's actually a really good question, and it's a big limitation of what we do. Like, so you can't, rats aren't good patients. They don't like doing their PT for some reason, and I don't know why. They're just kind of jerks that way. So people try to encourage them to be active by like enriching their environment, putting toys in there they might want to play with, things like that. But really, the problem with spinal cord injured animals is rats are social animals, but when they're paralyzed, you can't house them together because they'll chew on each other's feet and eat them off because they can't feel it and it's really gross. So you actually discourage them from having activity. So there's ways that you can try to overcome that. And that's like a big factor of what we're doing next is having these more stimulating environments for the animals so that they are encouraged to have more activity. But the reality is it's suboptimal. So we're probably not able to elicit as much functional recovery as we might otherwise, because that's a pretty big limitation.
Thank you. We're gonna clap now as a clap.
Well,
thank you so much for making the time.
No, thanks for the invite, and I appreciate you guys listening to me yap for over an hour. I didn't realize how long I went.
No, that was get myself back on here. That was great. And I may relay if people have some after the fact questions. I may like send you some correspondence that I could share with them or.
Sure.
Okay. All right. Well, thank you so much for taking the time to do this.
My pleasure. Thanks, guys. Bye, Mark. If you're still there.
He's there. He's waving. All right. I'll see you.